Good morning, everyone. I welcome you all to IITR Diamond Jubilee Elocution Address, that is IDEA series, for today's lecture by Dr. Narayana on conventional toxicology needs, a review for naturals, especially those with HOSU. I would like to call upon stage our Director, Sir, and Dr. Anant Narayan, Sir, on the stage, please. For our today's lecture, Dr. D.B. Narayan. He is a pharmaceutical scientist with over 40 years of experience in research and development. He is recipient of Eminent Pharmacist Award of Indian Pharmaceutical Association in 2007. He has worked in both pharmaceutical industry and food industry, apart from leading R&D on botanicals and Ayurvedic products. He has contributed to monograph development for botanicals in IP. He championed regulations for notification of pharmaceutical as well as nutraceuticals regulation and recently Ayurveda Ahar regulation in India. Demonstration of reverse pharmacology approach with commercially successful products has been his forte, apart from leading research for several anti-cancer drugs, first time in India. He has set up Ayurvedic Trust in 2009 to support research, conduct training courses, and publish papers, books, and articles, apart from giving inputs to government on policy and regulation. Trust has conducted training program on scientific writing, patents, development of quality monographs for herbs and Ayurvedic products. Microscopic identification of powered herbs technology has been developed in collaboration mode with Hamdard University. He currently chairs committees in Indian Pharmacopoeia Commission and in Food Safety and Standards Authority. He has been awarded IRF IPA Lifetime Achievement Award 2021. Welcome you, sir.
Once again, I request the director to formally welcome our guest of the day idea series with the book. Thank you, sir. Now I request our idea series, lecture series for the day to present your wisdom and thought process of our wonderful name of idea series. Thanks.
Face for next slide or for the next. Okay. Or I can use it.
Oh, you can use this. The arrow. Like this.
Okay. This is better.
This might be possible.
Yeah. मातृभ्यो नमः पितृभ्यो नमः गुरुभ्यो नमः आचार्येभ्यो नमः सर्वदेवताभ्यो नमः. I'm very happy to be here in front of this August gathering, and I'm a bit nervous today. I've never been nervous when I give talks. I don't know how many talks I must have given in my life till today. I fondly remember having given a talk here many years ago when I received Zaidi Memorial Award. So ITRC, I'm one of the recipients of Dr. Zaidi Memorial Award. And to come to CSIR, I think it's almost like my second or third home. I've been involved in CSIR. I've been honored, rather, I should say, to serve on a lot of CSIR scientific advisory committees. Now I think it's called RCs, perhaps. And I think on 2 terms in ITRC earlier, I was a member of SAC, and I had the privilege of interacting with a lot of scientists. Primarily, I am a pharmaceutical scientist. I am not an Ayurveda person, but having applied for almost 30 years the drug development and pharmaceutical science to botanicals and traditional knowledge based products, I have been learning and studying and doing research. So what I'm going to present today is based on few case studies and learnings of over 2.5 or 3 decades, but more so actual practical experiments that have been done in the last 12 years post my retirement. So I continue to do science and learning today, especially in the foods area, nutrition, and more so on the botanicals. And I'm very happy to be here. When Dr. Bhaskar talked to me last time when I came to meet him here, I had come for a CSIR meeting, AB and monitoring meeting. I met him. He said, we are having this Diamond Jubilee and we have an idea series. I said, ideas. Actually, I like always because ideas are very important. Ideas only when converted to becomes innovations. And therefore I said, okay, during winter I will not come. And so March we decided, and I am here today. And secondly, I must place and record here. It's been a very big pleasure and a lot of learning to have worked with Dr. Bhaskar, especially in his former stint at Foods Authority as advisor. He is a very conscientious scientist, and not just a scientist, but a scientific science administrator as well. He understands how to convert science into either regulations or innovations and what needs to be done to inspire people. He himself keeps abreast and he is generally up to date in any subject that he is dealing with. And he has led a large number of organizations. He has built some of them, and some of them are truly outstanding ideas which he has converted. Like, for example, IT FSCAN in Mumbai, the Food Training Institute, an excellent institute. And I don't know how many of you would remember. I think most of you would be the newer scientific group than when I used to come to ITRC. So I am going to talk about the topic conventional toxicology needs a review for naturals, especially those known with HUSU. And this is going to be a talk which will have some case studies and examples and some theoretical explanations of a totally new idea that I developed when I was working with Unilever. And I retired from Unilever Research in 2010. And thereafter, I've been continuing that thought process. So I'm continuing to do that. And I said I'm a little bit nervous because I'm going to speak to pharmacologists, toxicologists, and scientists here. I am afraid because I'm trying to carry a small drop of a holy water to Triveni Sangam. You're all Triveni Sangam here who are the experts in the pharmacology, toxicology. And therefore, I'm a little bit, but I hope it would stimulate you to think a little outside the box, like I have, and some of my scientists, we have worked together. And this consists of 5 parts of talk. Please don't look at the number of slides. They are very large in number, but some of them will just go through. So don't worry about the time, but I'm sure I will keep you glued to your ears, eyes, and brain, because I'm going to give you a little different way of thinking. What is HOSU? HOSU is a short form which reads as History of Safe Use. History of Safe Use. What is HOSU is going to be explained by me. And then I will talk about safety assessment and quality, especially of botanicals, naturals. I'm using the word botanicals because people use different terminologies, herbs, herbal medicines, herbal preparations, and there is no uniformity globally. So botanicals is a more common word which applies to everything. There could be botanicals in one basket which are totally unknown, new, not documented. And there are botanicals which have got documented in various traditional knowledge books. Then what is the new approach, and some case studies I will present. And then I will also take you through how to think alternative approach than just conventional toxicology. And I would like to propose some work that a premier institute like IITR should consider. And I would volunteer to work with you all guys. And what is the way forward? First part, HOSU. What is HOSU? HOSU, as I said, is history of safe use. Historically documented for its safe use is called HOSU. This is a word that we coined when I worked with Unilever. When I joined Unilever in 2002, I was faced with a very serious problem. Unilever wanted to use naturals and botanicals in their products, both for oral consumption and for external use. But Unilever is a company, globally, they don't do any testing on any animals. So how do I test toxicology or toxicity or safety of a natural? And there I started looking at in a very different way. We started looking at it in a different way. So after 2.5 years of in-depth study, we developed what we call as a fishbone diagram to assess safety of naturals or botanicals known in tradition. And that is now a globally accepted policy across Unilever. No testing is done on any animal, and that process allowed India to get more than 35 herbs approved for safe use. And that time, one of the important things that strikes you, especially for you people, we should be able to differentiate between safety and safety profile. Nothing in this world is absolutely safe. We must know what is the safety profile of the ingredient or botanical that we are talking of. This difference is what we try to bring in Unilever. Today, history of safe documented use. The word documented use is very important. Is recognized by a large number of regulators. Traditional health, medicine and products directive in EU recognizes that if there is any botanical which has a documented history of 30 years of usage in any part of the globe and at least 15 years in any one of the EU nation, then you don't need to do toxicology, safety, and you can get a much abbreviated registration of that botanical product. The same is accepted even in Indian nutraceutical regulation. Only difference is in EU it is 30 years and 15 years in one of the EU country. In India it is 30 years global or 15 years in India. Experience of having used it or documented, and there is safety profile having been known. TGA Australia recognizes documented history of safe use and provides easier registration for botanicals. And SFA, that is Standard Food Organization Administration in China, State Food and Drug Administration also recognizes, because China, like India, also have documented history of usage. Most recently, botanical drugs, which have been approved as drugs, not as cosmetics, not as nutraceutical, not as supplements, and they have given recognition to his HOSU. So imagine the importance of HOSU. And Indian Drugs and Cosmetics Act, especially for Ayush, also do not ask mandatory safety assessment except in certain cases. So here are the three examples. In the last 3 years, 3 botanical drugs have been approved as drugs by United States Food and Drug Administration based on a guidelines issued in 1996, which has been amended in 2012. And there are, you can go to the Google and get it down. Type guidance to industry for botanical drugs. You will get complete regulations for developing a drug out of a botanical. The first product was Veragen, And this is a standardized tea extract, standardized to cyanocatechins, made into a cream to treat genital warts. And because of the documented history of safe use, United States FDA gave them a permission to go to straight away phase 3 clinical trial, and no toxicology data was asked for. And in the phase 3 trial, when they got good result, they were given marketing authorization. Second drug is from a South African plant, red blood dragon tree, which is a cactus, which has been standardized to a compound called crofelemer. This is a oral drug to treat diarrhea induced by anti-AIDS drugs. AIDS patients take anti-diarrheal drugs, anti-AIDS drugs. Soon they start getting diarrhea because of the irritation it causes, and they stop taking anti-AIDS drugs, and that's one of the basic reasons for failure of a large number of anti-AIDS drugs. Because it is a non-specific diarrhea, you can't treat them with antibacterial or antibiotics or others. So this botanical specifically had history of documented use in South Africa. Again, straight phase 3 clinical trial and drug was approved. And the last one, which was approved in March 2024, is from an extract of birch. You must have heard of this plant called birch. It is not an Indian herb, but it used to be used in Indian Ayurveda also. And this has been standardized to triterpenes. And this is again externally applied to treat children for various kinds of epidermal diseases. Again, because of HOSU usage, documented, provided, straight away phase 3. Now you can understand the power of history of use. Here is a slide which tells you the documentation in Ayurveda. I only listed a couple of them and the year in which they were written. They are documented. Today also it is available. Some of these books translated to English or Hindi is available. So in each of these granthas, there is history of documented usage. Similarly, here is a slide which shows you the traditional Chinese medicine documents. You might ask me as a scientist, what are you talking? Are you talking of snake charmer, herbs and books? No. These books have been written after huge observational evidence of what they call as Anuman Pratyaksh Praman. Anuman means there is a lead. Somebody said Aptavachan. Aptavachan means somebody suggested you got cough. Why don't you take some pepper rasam or pepper? It will help you to improve your immunity and cough will go down. So when that was done, Vaidyas used to test it, making products out of it. And then when they got convinced them, they exchanged the thought with other Vaidyas, and so therefore multi-locational testing used to happen. And I am told in Ayurveda, at least 7 years of observation on the safety and efficacy used to happen before that can enter into one of the shlokas in the granthas. So therefore, it has gone through a lot of observational studies, and they may not have an RCDB kind of a data that we may want today, but they have gone through the whole testing, and that's the power of HOSU. So the question that scientists like you would ask will be, how do we prepare a HOSU document? And I have been working in this area for a large number of years. So we published a paper, How to Document HOSU in Journal of Ayurveda and Integrative Medicines. And I'm very glad to tell you that it has received something like 876 citations as on today. So many people are looking at it because there were no guidelines. So what do I mean by HOSU? I must explain to this scientific gathering. Traditional books are available in which botanicals would be cited. And what would be cited? Which part of the plant is to be taken for that particular usage? How to process it? Process is very, very important. Not just the name of the plant or part, but the process is very important. Then the dose and level of usage, duration of usage. Don't take it for more than 1 month. Don't take it for more than one mandala. One mandala is 48 days. If you have to consume it for longer period, traditional knowledge says give a break after 48 days for 1 week, and then again you can start again, because the body becomes resistant to that and it stops recognizing it as a potential drug or a supplement or a health benefit giver. So you have to reintroduce that to the body. And indications of safety are documented. These are very, very important. Then to document, you have to give references to these books and all those details. That will be the cause of the part of the documentation. Commercial data. Today, a huge amount of commercial usage data, both in India and export to various countries, are available. Especially, there is a website of the Ministry of Commerce and Industry and Department of Commerce. For every botanical, for every ingredient that gets exported in India or used in India, there is a code called HSN code. And this code, if you know, and if you have access to this website, you can generate for the last 15 years how many tons of that herb or that product has been exported, what is the value of that. This again gives you additional data that the product has been put into that market. And therefore, that is very, very important. And then bill of lading. Every company which exports anything, you will have a bill of lading, export documentation. Copies of them would be very strong data to show that this has gone to those countries and people have used them. Pack shots of products in good recognized scientific books or publications or catalogs. If a herb containing product is in that market and you trace it out from 10 years old magazines, they are also evidence that the product has been in the market that time and people have been eating. And normally what happens is, if there is a safety issue, people would represent or refer or report or stop using it. The sale will not happen. So that's again an evidence. Then published review. As scientists, you all produce scientific review based on published data. They add to value. So in the case of tea catechins, huge amount of scientific data on safety as well as biological activity of tea catechins and teas are known. You add that to the traditional knowledge, it adds. Like for example, you have heard of GRAS document, generally recognized as safe for flavor usage, or in case it is a microbe, there is a documentation called quality presumed to be safe, QIP or QPS document. These are all again history of safe use document. And I regularly refer to a contribution of ICMR. I really feel excellent book, which are series are published called Reviews on Indian Medicinal Plants. Excellent scientific reviews are available. So these all cause history of use documentation. I must give you an example. How does it help? 3 years ago, I had been to Germany and I met the Counselor General. They had invited me to speak to their herbal industry in Germany and Ayurveda industry. They all said that Germany does not recognize amla. Can you imagine that a fruit like amla and an amla containing Chyawanprash is not going to be allowed to be sold because it is not registered? I was really surprised. And I came back and I worked along with a friend of mine who is an expert in Ayurveda, Dr. Baba. We prepared a 16-page document showing how amla is a dietary ingredient. It has also medicinal properties. It is documented in our granthas. And these amla preparations are sold in India today as chutneys, as murabbas, as so many other products. And we try to calculate the tonnages of the amla consumption and exposure to public. How many grams of a pickle, amla pickle, would somebody eat, and how it can be calculated to the consumption pattern which you all toxicologists do. And then we approached Cefexil because I could not have access to export data, and they took out the data for as a special case and gave us 15 years data of export of amla and amla containing product to Germany specifically, both in tonnages and in number of euros or dollars. Armed with all that, we gave it to one of the persons in Germany, and in 6 months, amla is now registered, not with any toxicology evaluation, not with any efficacy studies, but primarily based on history of safe use. So you can imagine the power of history of safe use documentation. So this is very, very important. Having laid down this importance of history of safe use, how do we look at safety assessment and quality? Safety is related to quality of a botanical or any drug for that matter. So how do we look at this? So in a common way, if I or any applicant had put an application of a botanical for getting it approved as a nutraceutical or as a drug in India or abroad, immediately the first thing is, please give us toxicology data, 90 days, animal study, this, that. And is it required? That is the question that we raised when we were working in Unilever. And the result is a different way of thinking for assessing safety of botanicals. So this needs to review to differentiate between undocumented herbs and documented herbs. This is very, very important. So the first requirement is we must differentiate what is not documented and what is documented. I'll give you a couple of examples later. Once it is documented and we know, we need to compare whether our present batch for which we have applied or the applicant has applied is processed as documented in the traditional books, or it is completely different from the process given in the book. If it is completely different, then it may not carry the extrapolatability of the history of safe use. So therefore, we must differentiate documented and undocumented. So we must compare that. We can also compare with a botanical reference substance. Governments and laboratories like yours or CSIR or Ayush can prepare samples of botanicals, authenticate them along with the documented usage literature. They can say this is a botanical reference substance against which you can compare a commercially produced botanical extract or fraction of it. Or suppose one of you as a scientist is working on a botanical, you have assessed safety, you have also conducted a clinical trial or some studies on its efficacy, and you call that a biobatch. In pharma industry, we call it a biobatch, means which has been established or studied for quality, safety, and efficacy. You want to compare that with a present commercial batch. Something wrong can go when you produce commercially tons of that botanical. So you can compare. So you can also compare with any other present. So I would give you the baskets of documented and undocumented usage. Documented usage, I have already explained to you. They are cited. Part of the plant is given. Genus and species are clear in that. Identity is clear. Process is documented. Dose of usage is there and indication. What is not documented? The herb itself is not listed in any of the granthas. So undocumented. So it would need a fresh look. Even if it is herbs are cited, you want to use a different species of that herb, which is not documented in the grantha, that's a new herb. You want to use its substitute or adulterants, again undocumented. And you want to use a different part of the plant. It is not listed in the granthas, so you have to be very careful. Simple example, all of us eat tomatoes. Tomato has a safety profile, it's a nutrient plant. It is also used as a medicine in certain cases. But do you use tomato leaves? Do you use tomato roots? Do you make a vegetable out of it? You never do. Simple. Similarly, our brinjal. Brinjal is a fruit. We use it. It has a lot of health benefits. But do we use brinjal leaves? If you eat it, it is very toxic. Tomato leaves are not safe. So you must differentiate between the part of the plant which is documented and not documented. And then differently processed. there is a water extract or infusion or cold extract or powder, or treat it with, let's say, cow's urine, or boil it with milk and take kshirapak. There are processes given. If you make it as per that, the safety profile of that is known, safety has been assessed. You want to use a totally new solvent, in the Granthas, they knew at that time only alcohol and water and oil and ghee and milk and cow's urine. Some of these are the vehicles. Today you want to use an ethyl acetate extract, acetone extract, petroleum ether extract, butanol extract. You want to use. Are they documented? No, they are not documented. So you need to assess them. And therefore, new technology, new levels of usage. Higher levels of usage can give you toxicities. Simple example, I'll give you curcuma, our haldi, which is very, very effective and a huge number of benefits are known. Regular use of high level of even simple turmeric powder is not good. It has side effects. And therefore, you must recognize that doses and usage has to be respected, what is given in. What happens, you might ask me, if you consume higher level of curcuma, your blood will become thinner and bleeding time will not be controlled. So there could be several side effects of, and even higher level of curcuma for longer usage can cause liver damages because of certain other reasons. So therefore, the level of usage is also very important. You want to use it for an unknown new indication. Grantha or documented studies does not put that indication and you want to use it for a new indication. Again, it is undocumented and there could be other changes. So as I said, most botanicals, at least in India, China, Bhutan, Nepal, Malaysia, Germany, and some of the countries which have got huge documented traditional knowledge, they all have HOSU opportunities. And most botanicals have health benefits and they have also got therapeutic benefits. The difference between a synthetic drug and a botanical is botanicals are multi-component. They act on multiple organs concurrently. They may be working on multiple receptors concurrently. And it is very difficult to pinpoint which molecule in this herb is actually responsible for that particular biological activity. There can be many compounds that are present. And you all recognize more than 60% of today's synthetic drugs or NCEs have an origin in one or the other herb. Even Taxol came from a plant origin. So more and more drugs are coming out isolated as pure compounds. But what we are talking is not the pure NCEs, but the extracts or processed materials. And there is a huge amount of interest in scientists, drug industry, food industry, and pharmaceutical industry, as well as fertilizer industry to use more and more botanicals for benefits. And most often, pharmacopoeia's demand for quality, and like we are scientists, we all look at what we call as a marker compound approach. What is the background of marker compound approach? Western scientists, Western industry, Western regulators did not understand botanicals. And in 1994, when United States brought out the regulation, Dietary Supplement and Health Education Act, DSHEA, when it allowed without FDA approving any botanical, you could use it as a supplement or as a nutraceutical. That time, large amount of herbal products from China and India started getting exported to US and getting sold there as supplements and not as drugs. So suddenly Americans say, oh no, how about it? We want it standardized. So the pharmaceutical thinking said, we must look at a bioactive compound in this herb, which they gave the name marker. And they started isolating and purifying one or more compounds from the herb and calling it markers. And how do you say that is a biomarker? Nobody can do the testing on human beings. All that they do was some in vitro test or in vivo on some cell line or some organ tissues, liver tissue, pancreatic cells and cell lines. And if they found some activity, they declared this must be the one which is responsible. Soon they ran out of such compounds because most of them turned out to be not active even in those in vitro in vivo models. So then they came up with a new thought called analytical marker. There is a compound which is only good in analysis. I can analyze it in HPLC, GC or whatever technique, and they call it. So we came up with biomarker and analytical marker approach. Then everybody, every scientist, every commercial, every marketeer, every regulator thought marker compounds are the best to quality control. But are they correct? They are not correct because one compound or more than one compound is not going to tell you the biological whole thing of the whole entire plant. There are so many compounds which have got present in the plant. So we in our team, led by my thought process, did some studies, and this is a paper we published in Journal of AOAC International in 2019, where we looked at additional approaches other than purified single marker compounds. Are there other compounds which are active? We came across huge number of other compounds. In some plants, the sugar was active. In some plants, potassium was found to be very effective. Arjuna is very good for heart. Every scientist talks about Arjun janin as a marker. We tried to do some studies and found Arjuna bark and wood is the richest source of potassium. And that high source of potassium, which is in a complex form with organic compounds, are responsible to protect the heart and reduce the ill effects of sodium present in whatever food that we take. And in some other herbs, there are fibers. Fibers are prebiotics. Fibers have a lot of benefits. And in some other compounds were there. So there is a need to look at different approach. So if we take a botanical, most often what do we test? We test for chemistry of marker compounds, one or more bioanalytical. But look at the structure here that I have tried to put. Identity and physicochemical tests are very important. Tests for contaminants have to be done because sometimes the toxicity is only because of the contaminant and not the herb. I will give you an example. During Corona time, Ministry of Ayush promoted use of guduchi. You must have heard of this compound, Giloy, Amrita, or Tinospora cardifolia. Its stem or roots are used. And suddenly it started getting attacked saying it is causing liver injury. Whereas in the Grantha, guduchi is a panacea. It is an immunomodulator. It protects the liver. So I tried to evaluate and I found that guduchi is very rich in starch and carbohydrates. And if it has a little bit higher moisture, on the surface, lot of bacteria and fungi start growing. People don't clean guduchi, they use it as such. They boil it in water and take the decoction, or they take the guduchi powder. So you're also taking the fungi, which would contain aflatoxins, which would contain several other toxin compounds, which are contaminants. They are the ones which are causing the liver injury. So we got a very small study done. I am a one-man army today, having retired. I don't have funds from DST or ICMR or DBT or anywhere. So I do get some studies done. What I did, I took a sample of guduchi, cleaned the surface, sterilized it, removed all the fungi and made sure that it doesn't have any toxins contaminated. And that powder against an uncleaned material, I got some testing done. I found that it was the contaminant level was very, very high in the raw drug. And even if boiling, aflatoxins don't get destroyed if you just boil it with water. So that is the area people have to think. What about carbohydrates, protein, fats, and fibers? These are present in almost all botanicals. They have got a equally important role to provide activity. Example you want to ask as a scientist, same guduchi in Ayurveda, there are two methods of processing. One method of usage for Ayurveda for guduchi is they don't do any extraction. They just powder the guduchi root or stem, put it in water, large quantity of water, leave it overnight and keep stirring. Next day morning, you would see a whitish powder which is separated from the root powder and settled at the bottom. You decant that, filter it and wash it. All that powder that has settled at the bottom, which is nothing but carbohydrates and starch present in guduchi. And that is called guduchi sattva. One small bottle of 5 grams of guduchi sattva sells at 600 odd rupees. And the dose required is one ratti. One ratti is a pinch of that, which is equal to 125 milligram. You are given twice a day. And that is works wonders as an immunity provider. So starch and carbohydrates and fats and fibers are also important for biological activity as well as for safety aspect. Metals and minerals. I give you an example of Arjuna and the potassium role. Many herbs have vitamins, amino acids. Most commonly we take moringa. Moringa has become a very popular herb now. Shigru it is called in Ayurveda. It is the richest source of folates. Richest source of folates. And it is very good when you take it internally. And it has got bitter compounds which are very, very important. And these bitter compounds are nothing but aglycones of the moringa, and that has got anthelmintic activity. Simple. It has got folate, and that's why moringa vegetables are given to pregnant women and lactating mothers because it promotes lactation as well as it improves the hemoglobin level in the ladies. So moringa is very good. Are we talking of a marker compound there? No. We must look at it in a holistic way when we analyze. So therefore, in summary, just looking at marker compound and assessing is not a very good method. So therefore, when I joined D.B.A in 1991, we used to say we are having very good quality control of our herbs, and we used to just work on a good TLC. TLC can give you a fingerprint profile, and soon TLC was being questioned. Oh, what is this? You are looking at only some 5 spots in a TLC, 8 spots in a TLC, and you say you got great control. So they started a marketing war. People said, I will look at marker compound and TLC. Then this war started saying, if that guy gives you 25% of this marker compound, I'll give you 30%. Somebody said, I'll give you 60%. And so, is it responsible? You go on fractionating that herb, you are going away from the grantha. You have got a new extract, you are not worried about its safety, but you are fighting in the market saying, I will give you a higher level of that particular compound. So this is very, very difficult to say. Now, contrary to that, I would like toxicologists to think, do we consume the herb or the process as such? After processing, when we consume it orally, in the stomach, it goes into an acidic medium, and in the intestine, it goes into an alkaline medium. Many of the marker compounds, if you look at its careful study, in acidic medium, they would have degraded, or in alkaline medium, they would have degraded. So they may not get absorbed, their solubility gets altered. So we are again not looking at in a very holistic or a broad way, but we are looking at it in a very reductionist, what I can understand, I will do. I will not worry about the other part. So therefore, is there a consensus on marker compounds? Here is a paper, not mine, not ours, but published in International Journal Planta Medica, which looked at all the pharmacopoeias in the world for the same botanical, which pharmacopoeia has how many markers and how they are tested. And they have, and whether that gives a better control on the quality or safety of that botanical. So therefore, they questioned and they very clearly have written conclusion saying there is no consensus on whether a marker is good for quality and safety or whether more than one marker or how many markers are good. So there is no consensus today. So we need a new approach. This is exactly what we tried to do in Unilever in a little way, which I am now continuing. And how do I do? So we need to move away from incomplete, too much focus on marker compounds and a reductionist focused, what I understand, what I can test only, or what I will do. This is like the famous California proposition. I don't know how many of you have heard of California proposition. Lot of calcium carbonate and several other compounds used to get exported to United States. Somebody put a case in California court saying this calcium carbonate and this supplement that I'm eating has higher level of lead and mercury or higher level of lead and heavy metals, and they are giving me a problem. So the court asked who has prescribed the acceptable levels of lead. United States FDA, please underline this statement. All of us think that US FDA is God and gold standard. It is not. We in India are better than US FDA. We must recognize that. And United States FDA gave a statement to the court saying, we have not determined the safe levels or highest level for lead and heavy metals. So what does the court say? Court is what? He is not a scientist like all of us. He is not a toxicologist. He is not a chemist. He asked, how do you test for lead? They said we do, today maybe the answer would have been, we would do ICP-MS. Okay, how much can you detect? Then the company got stuck. So they said we can detect 5 parts per billion. Court gave a judgment, 5 parts per billion is the limit. All calcium carbonate and drugs sold in the market should be below that. Can you achieve that? To purify compounds or botanicals for heavy metals. So therefore, we need to look at it in a holistic way. We must try to coverage as much as possible. So in Unilever, my problem in Unilever as head of herbal research was to convince a white guy sitting in Denmark or Europe or in UK to accept the safety of my particular herb. How do I convince him? So therefore we said, can we demonstrate to you what we want to use is closely similar to what is the Hosu? Can we demonstrate? He said, yeah, that's a fantastic thing. Show me that. This is what we developed. And again, I go back to the same slide. What do we compare? We compare in any one of them. We compare whatever we want to use, what you want to use tomorrow with a Hosu sample. And therefore, we adopted a technique. We opened up our minds and said, why should I do only HPLC, HPTLC or metal? I will do all elemental, instrumental tests that I can do. I'm not blocking my head. I will open up my head. So therefore, today there is a technique called principal component analysis, which is part of an SPSS statistics software. And this helps you to plot similarity and dissimilarity based on the number of data points that you can give to the particular data. So here is a case one, which is a published paper. We took Terminalia cebula, our Haritaki, hardered. We took that. We prepared a Hosu sample exactly as given in Ayurveda. Take this, powder it, put 16 times water, boil it, concentrate it to 1/4, and then further concentrate it. Whatever has to be done exactly as per that. We obtained a commercially produced water extract, a commercially produced methanolic extract of Terminalia cebula, and we did all multiple analysis of these samples. And I'll show you the data. So we are comparing a Hosu sample with commercial or produced batches. And what all would we do? We need to enhance the data points. If we do TLC, we may get 5 spots, 8 spots, 6 spots, and that is very difficult, very less in statistical analysis. So we did IR, we did NMR, we did UV, we did UPLC, we did HPTLC, HPLC with two different mobile phases, HPTLC with two different mobile phases to separate different group of compounds. One looking at sugars, one looking at polyphenols, one looking at glycones, one looking at glycone. You can do that. And then look at mineral values, flame photometry. You can do anything. And we have published this paper. So we did all this. And when we determined known compounds in Terminalia cebula, this is the kind of data that we get, where we analyze for cebulic acid, gallic acid, chorioligin, TGG, cebulanic acid, ellagic acid, cebulanic acid. The data is varying. We are very difficult to compare the two. So here is the various analytical, this one, IR, UV, TLC profiles, NMR profiles of all the 3-4 extracts that I talked about. Then we plotted it onto PCA plot. It gives you a biplot which will tell whether our commercial methanol extract, alcohol extract, water extract, how closely it is similar to the HOSU. HOSU, why I am saying? HOSU means its safety is known, it is documented, published in scientific literature also. And we found that all the three extract, even though it was methanolic extract, was more than 70% similar in to HOSU. That means extrapolatability of HOSU safety can be done to these commercial batches. This is the first step. Second one, we said, how do I know that if there is a difference or if this method would work? We did a second testing where I got help from IHBT scientists on zebrafish toxicities. We took the same HOSU of Terminalia cebula. We did the similarity, dissimilarity, but one sample we fractionated it. The water sample we fractionated using acetone. So we have either removed some compounds or added some compounds. And then we put this to zebrafish toxicity because zebrafish toxicity method is accepted to be close to human beings and it is a well-known alternative methods that are used. And what's the result? We found that the acetone fractionated Terminalia cebula extract was showing slightly higher toxicities compared to pure cebulanic acid or HOSU material. It was not really very drastically different, but there was a little difference. In such a case, it is closely similar, slightly different. So maybe we should apply our mind as scientists and see what additional simple safety data should we generate to see whether one can use even the acetone fraction as to have a closely similar safety profile. So this is one method that I think scientists should consider for assessing safety both through HOSU and non-HOSU material. Are there any other innovative alternative strategies other than animal testing, other than toxicology? Here are some examples. I will give you an example of how to generate and evaluate. Several years ago, a pharmacologist took our simple tulsi plant and extracted and tested it on dogs and found that it was spermatogenic. It was causing death of sperms and it was also acting on ovum. And they said these are very, very toxic and they published a paper out of it. And so 8 European countries immediately issued a ban order on tulsi. Any product coming from India, they put some advisories and some of them they said tulsi can be problematic and so you ban it. So European companies tried, approached government. I don't know what happened. They approached me because I used to publish papers on botanicals. So they said, please help us, please help us. I said, please, this is to be done by the ministry and not by me. But they said, please help us. So I said, okay, I'll try. So I worked with Ayurveda people. What did we do? We took all the granthas, recipes in which tulsi is an ingredient. And we calculated in one dose or per day how much tulsi would be consumed if somebody takes this particular recipe in the Grantha. Today in the market, there are proprietary Ayurvedic medicines which are licensed by drug controllers and they are being sold. We took many of them and again calculated how many milligrams of tulsi would be present in that particular per day consumption. We selected some 25 experienced Ayurvedic Vaidyas who had 8 years, 10 years clinical experience. We just conducted an interview with them and documented it and asked them only 4 or 5 questions. Do you use Tulsi alone to treat your patient? Yes or no? Yes. How many grams do you prescribe them per day and what process? Do you ask them to boil it with water or eat it as such? What do you do? And then in your last clinical practice, have you observed any serious or other side effects of having prescribed tools? We documented. And we calculated the quantum, please underline, quantum of tulsi consumed. And when we compared the level at which this study was done on dogs, it was 1000 times higher than what is prescribed, even by clinicians. So if you are actually loading an animal with very high level of some ingredients, some botanical, you will actually get toxicities. We must evaluate it at that level and like toxicologists say, 1 or 2 doses higher than that just to challenge the particular testing. And remember, posology, the knowledge of calculating doses is entirely different from synthetic drug or pharma drugs and in Ayurveda. In traditional knowledge, even in traditional Chinese medicine and Bhutanese, it is not just the herb that is taken for matra is dose, but the ability of the patient to tolerate that dose is also considered in traditional knowledge. If the person is very weak, suffering from disease, unable to digest, he has gastric irritation, how can you give them such high doses of that particular thing? They calculate and slowly they start improving, increasing the dose to make the person react and give the benefit. So this is an example that we have to think outside the box. I did not go to the lab. I did not question that author. Why did you take this dose? And most importantly, toxicologists must always, whenever you do any testing of safety for a botanical, please consult a good Vaidya or a good Ayurvedic researcher. Find out what are the doses given to human beings and based on US FDA or Indian, calculate the animal doses accordingly and then perhaps do. Most papers have a reviewer in number of journals. People do toxicity studies and animal studies. Arbitrarily, they will say, we tested it at 100 milligram, 200 milligram, 500 milligram level. Arbitrary. Why did you decide that? Don't do that. Please calculate based on human dose. You have formula. Human average weight is so many kilos and animal weight is so many grams. You have got formulas. And I think that is very, very important to do. Another example of thinking outside the box. In Unilever, we had therapy centers, which is just like Ayurvedic Panchakarma clinics. And people had to be used with thylums to do massages, to do various other therapies. And again, unless we get it cleared from the Unilever safety clearance, we could not have used it. So can I go and do toxicity studies of those traditional oils that are used in therapy centers? So difficult. We said, why don't we go and interview patients on which these oils have been applied, and especially the therapists who do daily morning and evening different oils they apply and do massage and abhyangam and everything. Let us look at their hand, let us look at their face, let us look at their and ask them questions. They did cause allergy anytime, did it cause raising, did it cause inflammation? So then we photo documented 28 therapists who have used some 30 different oils. We photo documented very carefully their hand and documented their interviews and submitted it to Unilever. They said, this is the best evidence. You have shown that these are safe. people are using it and patients are using it and they are not getting any other side effects. So you can think in different ways innovatively. Of course, uh, we were questioned, does it cause any cancer, skin cancer? So we got various AIMS tests and other tests done on that, which are non-animal testing. So you can think of alternative, innovative approaches to evaluate or assess safety. So what's the way forward? Way forward is there is a need to have cross-functional interactions between traditional knowledge experts, pharmacologists, phytochemists, and toxicologists. That would give newer methods of looking at. And similarity and dissimilarity is one such example, what I explained to you. So next is need to recognize HOSU and develop scientific guidelines to reduce too much of animal testing. We must train our scientists to prepare HOSU documentation. And we have at least begun. Our paper may not be the ideal paper, at least we have begun. Please improve on it, read that paper, improve it, bring out newer methods, and learn to write safety reviews of botanicals based on published scientific data. At that time, I would recommend you must be very careful in selecting which paper should go in and which paper should not go in. This is very important because, like the example of Tulsi I gave, it is an irrelevant paper for that safety review. If you include, you have to say Tulsi will cause spermatal, harm and all that. But that is not correct. That is not at the relevant dosage at which it will be. It has to be reviewed. So be careful about what papers you have to include in the safety review. And need to review why testing is required. Most commonly, all toxicologists and pharmacologists say two species on animal में test करो. Why यार? The longest testing on human Homo sapiens have occurred. Why do you need on two species animal? Can it be reduced to one? Can it be reduced to minimum testing? That's a question that we should ask and generate and think about it. And also, if we can go through the botanical similar approach, similarity and dissimilarity, which I am calling bot similar. Today, regulators recognize biosimilar in case of complex biological materials. Why not bot similar? That should become a regulatory method for accepting safety as well. And in that case, if there is a little dissimilarity, question should be asked, what is the area of concern? Does that herb is going known to cause gastric irritation? Does it cause diarrhea or does it cause allergy? So only generate that particular area of concern data to strengthen our assessment. Also consider alternative approach. I have given you a couple of examples. And can toxicity testing in zebrafish, C. elegans, Drosophila, there are standard methods, and these are considered to be close to human toxicology or human methods. Can we use that to as an alternative testing to reduce huge conventional toxicity testing? I am now told that United States FDA in their new drug applications are accepting computational docking studies as part of safety assessment. If that is true, I don't know, please go and check yourself. If that is true, can we replace some of the conventional toxicity requirement with computational docking and also develop guidelines of what docking experiments have to be done, what should be looked at, what should not be looked at. Computational docking, please don't misunderstand me, is also hyped up beyond a level. Everybody is into that now, but it is relevant. If it is done in a particular way, it can add a lot of value. And develop a fishbone diagram using some of the thought process that I have said. And I volunteered at my own expense. I will come to IITR anytime you want, any number of times you want. If you wish to, please let us develop a fishbone diagram or a process. What do I mean by fishbone diagram? Here is a schematic diagram that United States FDA has published for guidance to botanicals. If you are looking at botanicals, when does it become a supplement? When does it become a nutraceutical? When does it become a drug? When does it become a thing that is undocumented and you need full toxicity? Similarly, can we prepare a diagram for that and adopting both similar as an approach? And I would be very glad to work with IITR on such a model if CSIR and IITR can take up under these Diamond Jubilee ideas. This is a totally new idea, in my opinion. I would be glad to do that. And I thank, in the end, Paul Russell, who is the scientist in Unilever with whom I worked, and scientists from Natural Remedies, Murali, Rojisan Koshi, Vinit Singh. We did these studies on Terminalia chabula. Director IHPT and Dr. Dinesh and Dr. Dhamanpreet of IHBT who did the zebra fish toxicity studies. And to all of you and to CSIR and IITR for inviting me to give this talk today. And your own former Director General Dr. Mashelkar used to say, umbrellas and parachutes work only when they are open. So keep your minds open and think of new ways. And today, India is at the frontier areas globally. And we must show to the world newer ways of assessment and botanical drug development and safety assessment, and not just copy what the West says. We should challenge the West. We are sitting on such huge amount of knowledge, and we must become leaders. And I would be very happy if even one scientist in the audience today thinks that one of the points that I made is worth doing something. Thank you very much. I would be glad to answer any questions if there is time. Is there time or no? Okay. 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 Please, no problem. And I am available here. Anybody wants talk to Dr. Kausar, we can meet and we can discuss further. Thank you very much.
Thank you so much, sir, for such an insightful talk. Now I request Dr. D. Kanuno, RC Chairman, CSIR IITR, to felicitate our guest with shawl and momentum.
Thank you very much. Thank you very much.
Thank you, sir. Now I invite Dr. Abhay Bajaj, CSIR IITR, to propose vote of thanks.
Respected Dr. Narayana, CSO at AYURVIDYE Trust, Director Sir, respected RC members, dear colleagues and students, on behalf of IITR, I would like to express my heartfelt thanks to all those who have made this event possible. First and foremost, I extend my deepest gratitude to our distinguished speaker for sharing your expertise and knowledge. Your presentation has been incredibly informative and engaging, offering us interesting perspective and ideas on HOSUs. Thank you, sir. We are grateful to you for accepting our invitation and grace this occasion. I'm also thankful to our director, sir, for his encouragement in organizing this event. Special thanks to Dr. Kauser, Dr. Partha and Dr. Nandita for coordinating this event. On behalf of organizing committee, my heartfelt thanks to all the scientists, students, and all the attendees for your active engagement and enthusiasm. I would also like to thank the technical support team and the staff of IITR for facilitating this event. Thank you very much.
I request everyone to please rise up for national anthem.
Thank you. हाँ मेरे